Thanksgiving. Let's go ahead and jump into chapter 34. My hopes is to complete chapter 34 by Thursday, so that next week all you have is just your exam. A week from this Thursday, and it'll be the same format, multiple choice, okay? All right, chapter 34, the origin and evolution of vertebrates now. So it says about 500 million years of backbones is what they say. Early in the Cambrian period, about roughly 530 million years ago, an astonishing variety of invertebrate animals inhabited the Earth's oceans. But it's believed that at least one type of these invertebrates gave rise to the vertebrates, one of the most successful group of animals. Okay, the term vertebrate comes from bones called vertebra. Okay, some people say vertebra. Which is a series of bones that make up your backbones. And these bones actually protect an organ called your spinal cord, which runs through these bones. Now, it's believed that vertebrates colonized land approximately 365 million years ago. And there are roughly 57,000 species at the moment of vertebrates, including the heaviest and the largest ones, which are the dinosaurs and the blue whales. Now, vertebrates vary significantly in their characteristics, or more specifically, their traits, such as body mass. So what we'll do as we go through this chapter is we'll start off with the basic body plan applied to all animal species classified under the phylum Chordata, except some of these species are actually invertebrates, but they do have the basic body plan of all chordates. Then from there, what we'll do is we'll divide the Phyla Chordata into 11 subphylas, starting off with the 2 invertebrate phylas, and then we'll work our way down to the remaining 9 vertebrate phylas. And we'll start adding on traits to that general body plan we start with, okay, for the chordate body plan, as these animals become more and more complex. So one of the earliest known vertebrates from the Cambrian period, which is roughly, if my eyes aren't deceiving me, which is roughly right about here, in this era of this eon, if you recall, we talked about the history of the Earth, okay, is the, if I can pronounce this correctly, the Milocomengia Fingawa. I've practiced that so many times. It's about 3 centimeters long, and it's one of the earliest, if not the earliest, vertebrate fossils that was discovered. Okay, so when we talk about these vertebrate phyla, or these subphylas that fall under Chordata, okay, we start off with aquatic species, and then we slowly work our way to terrestrial species. So that'll be something else I'll try to continue to point out. So as I said, from chapter 32, we proposed 2 hypotheses were proposed. I said we're following the second hypothesis. So if you look at what I have, some key parts highlighted, such as bilateria, deuterostomia, and based on the second hypothesis, deuterostomia consists of 2 phylas, phylum Echinodermata and the phylum Chordata. Now, chapter 34 is going to focus on this phylum here. Trying to do it all with the circle, make a little symbol here. Okay, this phylum Chordata. So the next slide I'm going to show you is going to highlight the bilateria and deuterostomia. Then I'm going to show you another slide that is going to not only show you deuterostomia, but going to give you details for the phylum Chordata. Instead of showing you Echinodermata and phylum Chordata, it's going to show you Echinodermata, and then it's going to show you the 11 subphyla. So keying in on bilateria and deuterostomia, that's what's shown here on this larger one here. You can see how these overlap with each other. Now for chordates up here, chordates that fall under the phylum Chordata, they're all bilaterian animals, thus they're classified under this group or subclade, that belong to a clade of animals known as Deuterostomia. If you recall, we talked about 2 types of embryonic development, protostome and deuterostome development. Now, chordates comprise all vertebrates, but it also consists of 2 invertebrates, the Urochordates and the Cephalochordates. So for some of these phyla, I'll give you their lay terms, okay, what they're more commonly known as, because many students are familiar with the lay terms rather than their technical terms based on the phylum. So the next slide I'm going to show you, okay, for deuterostomia, that's now shown over here. Okay. And it said Deuterostomia, based on the second hypothesis, consists of two subphyla. We have our phyla, the Echinodermata, but instead of showing you the phyla chordata here, this is something you're going to have to get used to in this chapter. Instead, they show it to you over here, and they're trying to tell you these are the 11 subphyla that fall under the phyla chordata. So they're giving you much more detail than the previous slide I showed you 2 slides ago. Okay. So under Deuterostomia, it's made up of two phyla, Echinodermata based on molecular evidence, and our phyla chordata. But instead of showing you phyla chordata, it's showing you the 11 subphylas that make up the phyla chordata. Cephalochordata, Urochordata, Myzini, Petromyzonatida, Chondrichthyans, Actinopterygii, Actinistia, Dipnoi, and then Amphibia, Reptilia, and Mammalia. You'll see this slide more frequently, more often, because it shows you some highlights. Such as there'll be a point once we cover the 2 invertebrate phyla, which are the 2 listed at the top. Then we'll start moving into our vertebrates, our phyla that are considered our, I guess you could say, our true vertebrates. We're actually starting to see the development of a spinal column. Okay, and it listed over here, and it listed over here. Then we'll start getting into phyla where there's a discernible or noticeable jaw, beginning with our Chondrichthyans. Okay, any vertebrate that has a discernible jaw, one that's noticeable, we call those jawed vertebrates Gnathostomes. So I'll talk about Gnathostomes. Then we'll get into where we have our first vertebrates that actually have, if you want to call it, true bone tissue or a lot of tissue contributing to their skeletal structure, beginning with this group, our Actinopterygii. So we'll talk about a mineralized skeleton, and we'll talk about how these groups, subphyla, are often put under a general label called our Osteichthyans. The oste, the O-S-T-E, that means bone. So you take an A&P class, you'll hear the term chondra, having to do with cartilage, and you'll hear the term oste, having to do with bone. So these are prefixes and what they mean. But then eventually what we'll start talking about are fishes that are called ray fin fishes, okay, and fishes that are called lobe fin fishes. And when we get into lobe fin fishes, we'll talk about the difference between the fin makeup in a ray fin fish versus a lobe fin fish. Because it was discovered lobe fin fishes, they have fins, but the fins have muscle tissue extending out into the fin. Therefore, they can do a little bit more with their fins. Then eventually we'll focus on this phyla, our Dipnoi. In lay terms, they're known as lungfishes. I added in a couple of links, videos. I forgot to check them to make sure they still work. Hopefully one of the 2 still works. And what they'll show you in these videos is how these Dipnoi are lungfishes. Because they're lobe fin fishes, which have muscle tissue innervating the fins, they can actually use those fins to crawl along the ground. Okay. Then we'll eventually get into how we have the evolution of vertebrate animals or animal vertebrates, okay, that are referred to as tetrapods. The term tetrapod is defined as vertebrate animals that have limbs with digits. Once we get to these tetrapods, which are animals that have limbs with digits, well, that's where we'll talk about our amphibia, reptilia, and mammalia. However, out of these 3 subphyla that are referred to as tetrapods, we'll talk about how 2 produce what is called an amniotic egg. Thus, they're often grouped together as amniotes. Okay. As we go through these 11 subphyla, starting with the Cephalochordata all the way down to Mammalia, okay, try to pay attention to the complexity of the animals. Okay, in other words, as we go down these subphyla, vertebrate animals, okay, are evolving more and more, becoming more and more complex and more capable, okay, of, performing more tasks, being more aware of their surroundings, the ability to manipulate those surroundings as well. So as I said, the Phyla Chordata, which chapter 34 focuses only on the Phyla Chordata, which consists of 11 subphyla. I said 2 of those subphyla actually consist of invertebrates. So then the question is, Why are those two subphyla placed under the phyla chordata if it's supposed to focus on vertebrates? Well, one is molecular evidence. We're following the second hypothesis, okay, which the phylogenetic tree is created based primarily on molecular evidence. The second reason has to do with what we call the chordate body plan, okay? Even though there are species classified under classified as that are invertebrates that are classified under the phyla chordata, what was discovered is that these species still have more in common with vertebrates than they do with other invertebrates. Okay, so like I said, part of it is molecular, but the other reason is actually in the phenotype, the chordate body plan. Any vertebrate animal classified under the phyla chordata, okay, is going to have 4 key traits that make up the general chordate body plan. At some point during their life cycle, they will have a discernible or detectable notochord. At some point during that chordate's life cycle, it'll have a discernible or detectable dorsal hollow nerve cord. Again, at some point during its life cycle, the vertebrate animal will have pharyngeal slits, or what they call pharyngeal clefts. And then at some point during the vertebrate animal's life cycle, it will have a muscular postanal tail. So it says here, the chordate body plan, all chordates possess the 4 highlighted traits at some point during development. In other words, at some point during their life cycle. They don't have to maintain this trait throughout their life cycle, but they will have it at some point in their life cycle. So they give you the simplest diagram they can for any vertebrate animals or any animal species that will be classified under the phyla chordata. Okay. Back here is the dorsal hollow nerve cord. You know it better as the spinal cord. Here is a notochord. Okay. Most of the time you'll hear the notochord being that part which evolves into our vertebra, our backbones. Then we have our pharyngeal slits or clefts. In fish, we call them gills. In us, it's our ears, our nose. Okay. And then as it says, a muscular postanal tail. Well, many vertebrates lose this, okay, during development. But what they mean by muscular postanal tail is posterior to the anus, you have a tail that has muscle tissue in it. Therefore, this helps with movement here. So as I said, the first trait for the chordate body plan, we call it the notochord. Chordates are named for skeletal structures called notochords, which they show you here just beneath the dorsal hollow nerve cord. Okay, the notochord is present in all chordate embryos and in some adult chordates. Now it's longitudinal in size or structure. It's flexible. Okay, and as I said, it's located just beneath the dorsal hollow nerve cord and the digestive tube, which is down here, this part right here. Now, it provides skeletal support throughout most of the length of the body. So there you can see they show you the notochord running most of the length of the body. And in most vertebrates, a more complex jointed skeleton develops, and the adult retains only remnants of the embryonic notochord. So as we go through these 11 subphylas, okay, there's a point where it's located in between the dorsal hollow nerve cord and this digestive tube here. There's a point where the notochord basically starts growing to the periphery or lateral to the dorsal hollow nerve cord. And then there's that point where it's what we simply call our backbones, our vertebra. Now, for the dorsal hollow nerve cord, I don't know if I said this on the recording for chapter 32, day 1, when I posted the recording for y'all to listen to and I canceled class. We talked about embryonic tissues known as germ layers. Okay. Blastic animals. Okay, we have ectoderm and endoderm at least. And I don't know if I said it on that recording, but for the ectoderm, it not only forms the surface of the animal's body, okay, such as our skin, but it also is known to form the nervous system. So if you look here, the nerve cord of a chordate embryo develops from a plate of ectoderm that rolls into a tube dorsal to the notochord. The nerve cord develops into the central nervous system in more complex animals. Okay, well, the CNS of the human body, or for mammals, is the brain and the spinal cord. These two organs make up the CNS. So here it's highlighted in the dorsal hollow nerve cord. But again, as I said, as we go through these 11 subphyla, we start off with very simple chordates as far as body plans go. They just have these basic 4 traits. But then we're going to add traits on as these vertebrate animals become more and more complex. So we start off with 4 traits with a chordate body plan, but then we're going to add on more traits. The third trait for the chordate body plan is what we call pharyngeal slits or clefts, which they show you right here along the lateral border here. It says the region posterior to the mouth is called the pharynx. If you're not familiar with this, the opening, our mouth, okay, is connected to what we, in our body, we say throat, but the throat is actually known as the pharynx in A&P. So they're roughly saying right in here we have our pharynx. Okay. Well, in most chordates, grooves along the outer surface of the pharynx, called pharyngeal clefts, will develop into pharyngeal slits that open into the pharynx. This allows water entering the mouth to then pass out of these pharyngeal slits. Okay. Now, depending on the type of chordate, okay, these slits can function in suspension feeding, in other words, allow for filter feeding, and or these slits can play a role in gas exchange. So in fish, we know them simply as gills. Now in tetrapods, okay, in other words, vertebrates with limbs or animals with limbs and digits, which are our reptiles, our amphibians, and our mammals, the pharyngeal slits were found to develop into the ear, parts of the ear, the head, and the neck. Okay, so again, chordate body plan is a basic, consists of 4 traits found throughout all chordates, but obviously over the life cycle, the structure changes. It's not so simple. Then we have a muscular postanal tail. It says chordates have a tail posterior to the anus. In many species, the tail is greatly reduced during embryonic development. And the tail contains skeletal elements and muscle tissue. And for those that retain this muscular postanal tail, it serves as a movement. It helps propel them or create force to propel the aquatic species, such as our fish and things of that nature. So again, keep that in mind for the Phyla Chordata. Even though 2 of the subphyla that fall under the Phyla Chordata consist of invertebrates, as are shown here, okay, you have to keep in mind the ones that are classified under the Phyla Chordata is based on the chordate body plan, those 4 traits. So these are the 11 subphyla we're going to go over. I'm going to go over part of these today, and I'm going to go over the rest of them on Thursday. Now, we'll start off with our 2 invertebrate phyla, known as our Phyla Cephalochordata and our Phyla Urochordata, but I tend to say subphyla. Then we're going to add on to the chordate body plan. We're going to add on additional traits to the body plan for vertebrates, okay? And then from there, we'll pick up with our subphyla Myzini and our subphyla Petromyzonatida. But after that, we're going to add on more traits, because beginning with the subphyla Chondrichthyans on down, this is where you see a noticeable jaw in the species. They've developed the noticeable jaw, so we're going to add on more traits to that chordate body plan that are going to be known as Gnathostome traits. In other words, traits associated with discernible jaw. And then we'll just keep doing that as we go on down. So the first subphyla under the Phyla Chordata are Cephalochordates. Okay, they're more commonly known as lancelets. Okay, they get their name from the shape of their body. Now there's actually approximately 30 species that fall under this phyla, and they're named for their blade or lance-like shaped bodies. Okay, now these guys go through what we call incomplete metamorphosis. Okay, as a larva, they already resemble some of what they're going to look like in the adult stage, but they develop a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a postanal tail. So during their larval stage or their juvenile stage, they go through incomplete metamorphosis, but they have all 4 traits of the chordate body plan. Now they are marine suspension feeders that retain characteristics of the chordate body plan as adults, so it doesn't change. That's why I say it's incomplete metamorphosis. And they're considered the oldest of the chordates under the Phyla Chordata. Okay. Now, So for these guys, they retain the 4 traits of the chordate body plan. This is going to be in contrast to our second subphyla, our Urochordata. They're also technically invertebrates, and they are more commonly known as Tunicates. This is a tunicate here. Here's a whole group of Tunicates. Now, for this phyla, this subphyla, our Urochordates or Tunicates, okay, they undergo complete metamorphosis down our larval stage and what they look like in their adult stage. You can see they undergo complete metamorphosis. However, notice here, in their larval or juvenile stage, they're pointing out that you have all 4 traits of the chordate body plan present, versus the adult stage, only one of those 4 traits remains. It doesn't matter, as I said. If the species at any time during its life cycle exhibits the 4 traits for that chordate body plan, we pull them under the phyla Chordata. So tunicates is what we call them. Here's a single tunicate here. They are more closely related to other chordates than our lancelets. Okay. Tunicates most resemble chordates during the larval stage, which may last only a few minutes. They have a notochord in the tail during the larval stage, but they lose those because they go through complete metamorphosis into an adult. Now for the tunicate, it's going to draw water though. It retains the pharyngeal slits, okay, and that helps it feed because it'll draw water in through an in-current siphon, which allows water then to pass through the gills so that they can filter feed, and then it'll exit through an ex-current siphon. So this is mentioned to explain, hey, this is why they retain this 1 out of 4 traits of the chordate body plan when they undergo complete metamorphosis into the adult. Okay, it's because they need to feed. Now, notice something, how it says the tunicates are more closely related to chordates than are the lancelets. But you have to keep in mind, okay, we're following the second hypothesis. So what I'm not highlighting is any molecular evidence also. That would also play a role. Okay. So then we've covered 2 of 11. These first 2 are actually invertebrates, but they both subphyla consist of, okay, species that have the 4 traits of the chordate body plan at some point in their life cycle. So thus they're classified under the phyla Chordata. However, it's with this third subphyla, Myzini, okay, that we actually start referring to them as true vertebrates. Okay, the catch is the vertebra or the vertebra-like structures they contain are actually made up of cartilaginous tissue, technically not bone tissue. Okay. Oh, I forgot to mention one other thing for these other 2 groups. Obviously, you can see in the larval stage there's a noticeable mouth, but in the adult stage of the tunicate, there's not a noticeable mouth or noticeable jaw or anything. Even here for the larval stage, you can see a mouth, but there's not a clearly defined jaw. Same thing for the lancelet. You can make out the mouth, but there's not a clear defined jaw. Okay, pointing this out because this is one of the traits we're going to be focusing on as we go through these phylas. For these 2 phylas, Myzini and Petromyzonatidae, what I'll point out eventually is they also have no discernible jaw, but you can definitely make out they have a mouth. So before we get into them, vertebrates are chordates that have a backbone. During the Cambrian period, a lineage of chordates evolved into vertebrates, and the vertebrates have a more complex skeletal system, and which was found they also have a more complex nervous system. They became more efficient at 2 tasks, okay, playing the part of predator and capturing food, but avoiding being eaten. In other words, avoiding playing the part of being the prey. They got better at those 2. So beginning with the subphyla Myzini, we add on to the chordate body plan. So I put in addition to the chordate body plan, we now add on traits common to the remaining 9 subphyla because those are the ones that are considered our true vertebrates. Okay, here's where we start inserting our molecular data. The first trait has to do with the Hox genes. I mentioned the Hox genes in chapter 32. These are referred to as genes that control developmental genes. And in chapter 32, I gave you all a slide, and on the recording, I had some figures on there that pointed out, did some comparisons. I believe it was a mouse compared to a fruit fly and a human compared to, I think, a fruit fly. And it had a bunch of little red boxes and everything. And if you listen to the recording, what I was trying to point out was that the little boxes that were all colored differently represented the Hox genes. And what was discovered is the more complex the vertebrate animal is, the greater the number of Hox genes, as well as different types of Hox genes, you will find making up part of its genome. So what they discovered is beginning with the subphylum Myzini, and on down for the remaining 9 vertebrate phyla. Okay, you have a second gene duplication involving the Hox genes. In other words, the number of Hox genes in these species that fall under the remaining 9 subphyla, they're more complex, and there's a greater variety as far as different types. Therefore, that contributes to more and more complex development. Also, we have the vertebrate enclosing the spinal cord. In other words, we're talking about the notochord wrapping around the dorsal hollow nerve cord, creating that spinal column with the spinal cord running through it. Another trait we see added onto the chordate body plan is the development of a skull, okay, enclosing the brain, which the dorsal hollow nerve cord, if you recall, I said it not only in more complex species develops into the spinal cord, but it develops into the brain, the 2 parts that make up the central nervous system, as well as contributing to the formation of the head. So we now start to get a discernible head being formed. And then for aquatic forms of vertebrates, we have the evolution of what we call fish, but for biologists or scientists, some fish they refer to as fin ray fish. Okay, and I'll explain that later as we go through it. So we have fin rays in the aquatic forms. They'll have fins, okay, and the fin ray part is the ray part is referring to they'll have rod-like bone structures that radiate from the lateral part of the body out to the tips of the fin, so they can be used like paddles or oars to help with maneuvering. So we add these 4 traits on to the chordate body plan for the remaining 9 subphyla. So beginning with these next 2, I pair these up, and the textbook pairs them up. Now the reason why we pair these 2 up, I'm just going to jump down to it, is these 2 subphyla, the phyla Myzini and Petromyzanus Tita, they're paired up for 2 reasons. 1, they're cyclostomes, and 2, they have a rudimentary vertebra. Okay, so these are the 2 key reasons why we pair them together. Now, the subphyla Myzini, they're better known as hagfishes, which are down here. Okay, there are roughly 30 species. The Petromyzanus Tita, this subphyla has been vertebrates whose members lack jaws. Okay, so they lack jaws, and that comes down to right here. It says not only has molecular analysis provided support, okay, that these 2 subphyla are vertebrates, but there are sister groups that comprise the jawless vertebrates called cyclostomes. So the cyclo, they say, is in reference to the shape of their mouth. It has a round shape. You can't see it here for the hagfish, but I posted a video for it, and I'll show you the link on the next slide. Now, rudimentary vertebrate, okay? If you go up here first, technically speaking, the various species of hagfishes or Myzini and the various species of lampreys or Petromyzanus Titas, okay, technically speaking, they don't have what we call a true backbone. However, they're classified as vertebrates because they have what is called a rudimentary vertebrate. In other words, they have something that kind of looks like, okay, a vertebra or your backbones, but it's made of cartilage, and it doesn't, they've heard it described as it doesn't actually completely encase the dorsal hollow nerve cord. Okay, but again, as I said, between molecular analysis and having something that resembles something of a backbone or a vertebra, even though it's made up of cartilage, they classify these, okay, as not only under the Phyla Chordata, but they actually classify them as vertebrates. So the lampreys here, I'm used to seeing lampreys either in a comparative anatomy lab where we dissected them. The only other time I've seen lampreys are on documentaries about sharks where they're swimming around the gills feeding on, food source on the gills. Here's the hagfish here. I gave you 2 links on YouTube. Just look at one of them. The hagfish, they have a way of defending themselves. Okay, they basically have what I call slimy mucus glands throughout their body. So that if a predator grabs onto them, or in the case of one of the videos, somebody grabs onto them, these glands will secrete this clear, slimy, mucusy, thick mix. So one of the videos, a person's handling them and he's showing all this slimy stuff, clear slime that's coming off. And on the other video, it's actually a research video where they're showing how predators will come up and try to feed on the hagfish. And part of the video will show a shark biting on the hagfish. And then all of a sudden the shark starts shaking left and right trying to get rid of the hagfish because it secretes that mucusy mix throughout its body. But see if you can take a look at those 2 videos there. Hopefully the links still work. I forgot to check them. Okay, before we go into the 7 remaining subphyla of the Phyla Chordata, okay, we need to talk about with bone tissue, or the evolution. So early vertebrate evolution. Okay, as I said before, the earliest fossils of vertebrates, they date back roughly 500 million years ago. We're talking aquatic, not just terrestrial. This included what we call the conodonts, which they're showing you one here. Okay, they're slender, soft body vertebrates, and they don't have a discernible jaw. Okay, so they're said to be without a jaw. And they had a skeleton that was made of cartilage. However, it was here in the mouth. They caught the attention of scientists. Okay, they have a set of hooks at the anterior end of the mouth that were found to be mineralized. In other words, one of the key minerals was just calcium. So bone tissue, okay, or the mineralization of tissue transitioning from cartilage to bone, it's believed to have first occurred in dental tissue. So far, that's what scientists have discovered, that's what researchers have discovered, that the first mineralized tissue that we could call bone, okay, actually occurred in the mouth. It was dental tissue. Now, the hooks that this conodon had were used to capture prey and move the prey into the pharynx, which we call the throat, where another dental element mechanically digested the food. Now, today, okay, those involved in the oil industry actually use the fossils from conodonts, their dental elements. They're used by petroleum geologists as guides to the age of rock layers, where they search for oil now. Now, I still need to search the background on how that works, but I do remember I had a student that was a geology major, and she was familiar with that. Okay. This here is just to point out something in relation to mammals, okay, and where they're also getting their, if you want to call it, their beliefs or viewpoints in. Mineralization appears to have originated with the vertebrate mouthparts, in other words, dental tissue. Now, vertebrates first developed a skeleton made of cartilage, and later this evolved into a skeleton of heavily mineralized cartilage. Now, where does this thought process come in? Well, we have those chordates that are invertebrates, or that are said to have, say, a rudimentary canal, but not true bone tissue. Yeah, that's what we're going to get to down here, right down in here, to answer your question, Catherine. Okay. So what it is, though, is that scientists know today, okay, that mammals in general in particular are a good example, okay, of species that during their life cycle, they will have what we call chondrocytes producing cartilage, as shown down here. And it's a known fact, say, in the developing human body, when the offspring is born, okay, that cartilage that is laid out by cells called chondrocytes will eventually be replaced with bone tissue by cells called osteoblasts. And you may have heard of this with newborns, where they tell you to be careful with the bones of the skull, okay, that they have not fully developed. In other words, they're still mostly made up of cartilage that is slowly being replaced with bone. So they combine with what they see today in present-day living species with what they're finding in fossil data to try and hypothesize what they believe to happen. Now, what causes mineralization forming the bones of the skeleton? This goes back to what you were talking about, Catherine. One hypothesis is a change in feeding mechanisms cause the mineralization of the skeleton, transitioning from cartilage to bone. Now, whether that meant that a primary food source disappeared and a more, how do you say, a tougher food source became available, and in order for a species to survive, they had to learn to, how do you say, consume that new prey. I don't know if you've had filet mignon versus liver, so sorry for the pun there. But also this led to the belief that mineralization began in the mouth and eventually the entire endoskeleton. So it's these two here. They believe it was feeding mechanisms. Yeah. If I have enough ketchup and onions to cover up the liver, that's good. But it's believed the bones did come into existence due to adaptations in natural selection, but natural selection, what specifically was it? Feeding mechanisms, the food source. Apparently there was a change in food source, and there were some vertebrate species that were able to adapt to that food source, and they believe that promoted the development of bone tissue or the evolution of cartilage to bone. Now, I put a video here. It goes under a type of what we call a process called ossification, endochondral ossification. Now, endochondral ossification, okay, is a process whereby cartilage is laid out by cells called chondrocytes. That cartilage basically serves as a blueprint for another group of cells called osteoblasts to come in, remove the cartilage, and replace it with bone tissue. Most of the bones of our body, okay, the bone tissue is formed through this process called endochondral ossification. But endochondral ossification, again, isn't just the production of bone, it's the production of bone replacing cartilage. So to that video, this also feeds into what they think was going on, or not going on, but that it's possible, okay, for cartilage to change into bone tissue or be replaced with bone tissue. Okay. So now what we have is the remaining 7 subphyla that fall under the phyla Chordata. These remaining 7 subphyla consist of vertebrates with jaws, which we call Gnathostomes. Okay, so it says here, jawed vertebrates outnumber jawless vertebrates. Well, jawed vertebrates or vertebrates with jaws, we know them as Gnathostomes. Gnathostomes include everything from your sharks to your mammals. Okay, now, if you look here, Gnathostomes have jaws that might have evolved from skeletal support of the pharyngeal slits. So, unlike the dental tissue, they believe that the discernible jaw formation of it, okay, originated from these pharyngeal slits, but not the slits themselves, the structural support surrounding the slits. So what they're showing you here in this region are the pharyngeal slits in this kind of clear color, blue color here. And then they're showing you some of the tissue, support tissue, which they refer to as skeletal rods here. They're highlighting a couple, and they believe some of this support tissue or skeletal rods eventually evolved into the bones of the jaw, such as the maxillary bones and the mandible, the lower jaw bone. And so this is what they're trying to highlight by color coding it. Okay, so bone tissue is believed to have originated from dental tissue, whereas a discernible jaw is believed to have originated from the pharyngeal slits. Now, some of the skeletal rods, as it says, evolved into parts of the jaw. The remaining pharyngeal slits remained as the major sites of gas exchange with the external environment in the case of aquatic vertebrates here. So we add on more traits, okay, and I forgot to write it down to the side, okay, but there are traits common to the Gnathostome body plan. Now you have to add these traits on top of the traits for the vertebrate body plan, on top of the traits for the chordate body plan. So we keep adding on more traits to the body plan because these vertebrates are becoming more and more complex, okay? Well, this is a Gnathostome body plan, so it consists of the 4 traits for the chordate body plan, the 4 additional traits that we added on for the vertebrate body plan, and now we add on a few more traits specifically having to do with jawed vertebrates. First trait, obvious one, formation of a jaw. The second one overlaps with one of the traits from the vertebrate body plan, okay? Genome duplication, or another way is we get an increase in the duplication of Hox genes. Then we talk about we get an enlarged forebrain associated with enhanced smell and vision. Okay. And in aquatic gnathostomes, there are organs that run the length of the body, the lateral part of the body, referred to as the lateral line system. The lateral line system is made up of sensory receptors that can detect vibrations, and it runs the length of the body. You see this in aquatic vertebrates that we just simply refer to as fish this very day. So you add this on for any vertebrate that is a jawed vertebrate. You add on these additional traits to that. But again, remember what I said. As we start with our first subphyla and we work our way down to our last subphyla under the phyla chordata, the vertebrate animals become more and more complex. Therefore, the body plan becomes more and more complex. So a little additional information for these gnathostomes based on fossil data in this case. The earliest gnathostomes include an extinct lineage of armored vertebrates called the placoderms, or plate-skinned. Most of them were very small, less than a meter long, but some fossils that have been discovered tell scientists that they could grow as large as 10 meters in length or more. They appeared roughly in the Ordovician period and became around 444 million years ago, but are believed to not have survived very long, but they don't know why. Okay, they survived till about 359 million years ago. Now, one of these placoderms, Dunkleosteus, it was known to grow up to 10 meters in length. And based on fossil data and reconstruction of the jaw, they believe that its jaw could create a force of up to 8,000 pounds per square inch, or PSI. So over here, if you're familiar with the Nile crocodiles, these crocodiles get very large, 20 feet in length at least. Their jaws can generate 5,000 pounds per square inch. So these placoderms here, the larger ones, such as Dunkleosteus, they were believed to have been had pretty powerful jaws. But that added to the fact that why was the time period that they existed so short? Because it wasn't considered very long. So here, what I keep giving you is this. If you recall, this is a geological record, just to give you an idea of where we're at. Ordovician period is right here, just a little more recent than the Cambrian period. So you can see we're working our way up as we go here, and it shows you their time frame here. All right, for fossil gnathostomes, okay, there's another group of jawed vertebrates besides the placoderms that they call the Gnathostomes. And they radiated. If you recall, we talked about radiation, so that's what they mean by radiated. They're talking about adaptive radiation. They radiated during the Silurian and Devonian periods, okay, roughly overlapping the time period of the placoderms. Now, by about 420 million years ago, these gnathostomes, okay, the Gnathostomes, diverged into three lineages of jawed vertebrates that survive today. These three lineages are known as the Chondrichthyans, the Actinopterygii, and the Sarcopterygii, except the Sarcopterygii consist of 2 of the 11 subphylas that fall under the phyla chordata, the phyla Actinistia and the phylum Dipnoi. Okay. Now, total though, these three here, as I'll talk to you about in a bit, they're also known as osteichthyans, because it's here with the Actinopterygii on down to the 11th subphyla that we definitely have vertebrate animals with bone tissue, extensive bone tissue, okay? But the reason why these two here, the reason why these two here are often put together as a third lineage known as the Sarcopterygii is because if you're not familiar with this, again, this is an A&P term. The term sarco, oh, I'm ready, it's just Tuesday. The term sarco means, you'll see sometimes where they say it means flesh, but often they'll tell you it's referring to muscle. So in A&P classes, when we talk about skeletal muscle cells, we call the membrane of a skeletal muscle cell the sarcolemma. We call the smooth ER of a skeletal muscle cell the sarcoplasmic reticulum. You keep seeing this sarco here. Sometimes you'll see this term in relation to cancer, deep tissue, flesh, or muscle, okay? So what I'm getting at is these two subphyla that make up the third lineage, Sarcopterygii, they call them lobe fins, not ray fins. These are the ones we're going to talk about that have muscle tissue that actually extends out into their fins. So they can do a little bit more with their fins than these guys called ray fins. Okay. So what we're going to get into is our first subphyla of Gnathostomes, our Chondrichthyes. There's just a little bit of information on these. Okay. As I said before, the chondra here means cartilage. So our Chondrichthyans, okay, they're basically fish. They have a skeleton, but the skeleton is primarily made up of cartilage. Okay, there are about 1,000 different species, and the two groups of species that are mainly pointed out and are most popular are sharks and our ray fin fishes. Okay, sometimes we tend to call them stingrays. And that's just a little bit of information I want you to know. I want to focus on these two groups. Okay. The second and third lineage, okay, that I was talking about, well, this subphyla, Actinopterygii, they're what we call our ray fin fishes. Okay, and it tells it down here too. These other two phyla, Actinistia and Dipnoi, as I said before, they form a group called the Sarcopterygii, and we call them our lobe fin fishes. Right here, they're trying to show you the anatomical structure of the fins between these two fish. We just call them fish. But you can see, okay, there's a different bone structure here for the fin than here for the fin. If you look here, you can see they're trying to make out bones that would be comparable to the human upper limb. So I'll go over this more as we go through these groups. So here it says the vast majority of vertebrates belong to a clade of gnathostomes called the Osteichthyes. So this is where we have our first true bone tissue making up the majority of the skeleton. Osteichthyes consist of both our ray fins and our lobe fins, both of these groups. Now, it says nearly all living osteichthyans have a bony endoskeleton with a hard matrix of calcium phosphate. Again, this includes our bony fish as well as our tetrapods. Now, aquatic osteichthyans are the vertebrates we formerly call fishes, just simply fish. So we don't tend to discern the difference between whether it's a ray fin or a lobe fin fish. We just say fish. Now, in general, speaking about both ray fin and lobe fin fishes, what they do have in common is that openings called gills or pharyngeal slits are covered by flaps of tissue called the operculum. For both ray fin and lobe fin fishes, they have an air sac known as a swim bladder. And for both ray fin and lobe fin fishes, just like the placoderm or gnathostomes, they have a lateral line system to detect vibrations. And they're also both oviparous, meaning that they lay eggs that will hatch outside the mother's body. So the fins are the key difference between the two, but they still have a lot in common. Therefore, we just say fish. Now, for the ray fin fishes, okay, fall under our phyla Actinopterygii, they include nearly all familiar aquatic osteichthyans. And again, the oste here, that means the prefix meaning bone. Okay, there are roughly 27,000 species of ray fin fishes. The ray fin fishes originated during the Silurian period, roughly 444 to 416 million years ago. They're not sure exactly when. Now, the fins themselves, okay, I'm going to go back over here to the one that I had before. The fins themselves for the ray fin fishes that are classified under the subphyla Actinopterygii, the fins are described, okay, structurally as being formed by bony rods that extend out, okay, from the base of the body on out. Okay. The fin itself is mainly just made up of these bony rods with the skin of the fish wrapped around it. Okay. At the base of the body is where you'll find muscle tissue that can move these, like if they're oars for maneuvering and turning and evading capture. This is in contrast to the low-fin fishes. The low-fin fishes, they have bony rods extending out, but also extending out, contributing to forming part of the fin, are bones with muscle tissue attached to them. So you can see here, they're showing you low-fin fishes. Making up part of the fin are bones. But what you can't see is these are bones that'll have muscle tissue attached to them, which you don't see here in the design of a ray fin fish. Therefore, low-fin fishes, and I have a video for one that you can look at later, low-fin fishes can use their fins as oars for maneuvering and evading capture by predators, but they can also, because of muscle tissue extending out into these fins, they can also use these fins to drag themselves along the ground, along the surface. Okay, and I have a video for one of them called the Phyla dipnoi, which are known as lungfish. Okay, and I'll show you a pic in a second. So as I said, for our low-fin fishes, they're called Sarcopterygii. The sarco is in reference to the muscle tissue that you find in their pelvic and pectoral fins. Okay, these low-fin fishes are believed to have originated or evolved during the Silurian period. Now, Sarcopterygii itself, it's the third lineage of the Acanthodians. Okay, and I said this third lineage is broken up into Actinistia and Dipnoi, but technically speaking, this third lineage also consists of the clade of tetrapods. Okay, but starting with the two subphylums in particular, there's not a lot to say for Actinistia. They're low fins. They more commonly are known as coelacanths, which were thought to have gone extinct 75 million years ago until somebody discovered one in the waters off the coast of South Africa. So here they're showing you a more recent picture of one off the coast of Indonesia. This is your coelacanth here. It's a low-fin fish. Okay. But it's this one over here that drew a lot of attention. The dipnoi, which also fall into this third lineage of Sarcopterygii. They're known in lay terms as lungfish. So I highlighted them right here. Here are a couple of different species of lungfish here. This one is in water, but notice this one's out of water. Okay. If you notice here, it looks like it's pectoral. This is its pectoral fin. It looks like it's bent, like it's caught on the mud. What you can't see is it's not caught on the mud. It's using its pectoral fins to help drag it across the mud. So I have a video here, and hopefully, again, I forgot to check them all. Hopefully it still works. It'll show you, okay, what these species are capable of doing. I don't know, I've never heard the term mud skippers. I've been looking at videos and everything. It may be. I don't call them mud skippers because you'll see on the videos they just drag themselves across. Maybe it is. I'll look it up and see. But these probably is. But these lungfish are dipnoi, under the phyla dipnoi. They're found in the southern hemisphere and are only found in freshwater. Now they're called lungfish because they have the ability, okay, to move out of the water and still breathe the surrounding air into their lungs because they have actually developed lungs. But they still have gills to help them breathe underwater. So they've got the option, okay? They can either use their lungs for gas exchange or gas exchange can be performed through their gills. Now, what else was discovered about these species that fall into the phylum Dipnoi, or these lungfish, is that during periods of drought, they have the ability to burrow into the mud. This one is actually coming out of the mud. What we didn't see is it actually burrowed into the mud, and it performed a process known as estivate. In other words, it went into a kind of hibernation, or it waited in a state of low body activity. They can actually bury themselves in the mud and remain there until the environment changes, okay, to a type of environment which is more beneficial to it. So in the videos, okay, they should show one that's coming out of this process called estivate. Okay, it's popping out of the mud as this one. it's using its pectoral lobe fins to help drag it across the ground as it's moving. So some individuals will say that tetrapods, including mammals, evolved from lungfishes. Scientists are saying there's a common ancestor between the two, but to say that humans directly or mammals directly evolved off lungfishes, they won't say that. They just say there's a common ancestor, okay, that evolved into our lungfishes or our phylum Dipnoa, and then evolved also into our tetrapods, which includes our mammals here. So there's a common ancestor here, but they have them right in here, okay, to indicate that, hey, look, these guys have lungs, and that's something these all have in common here. Okay, I'm going to stop here with slide 40. I'm going to stop the recording.